Okay guys, welcome back. In the last video we talked about what mitosis is, why our bodies do it, and we looked briefly at the steps. Now we need to go a little bit more in depth with the steps. And what I'm going to do is I've got your guided note sheet right here, zoomed way in. What we're going to do is focus on little bits of it at a time and fill it in as we go. So you can see I've got the interphase section right here. So actually I'll just scroll up right here so you can kind of get an idea of this is the notes that you hopefully already have. And then we're going to focus down here. So this little circle that I gave you is going to be our cell. We'll fill some things in there. And then this box over here is where you'll just write the name of each stage. So the first stage, stage 0, that we're going to be talking about is interphase. So I already have that written in there, so you don't have to watch me write that. So let's go ahead and start filling in what a cell looks like during interphase. And I'm going to be using 4 colors as we go through. The first one's going to be just kind of our main color, so I'm just going to use black, the color I'm already writing in. I'm going to use 2 colors that will represent my DNA, mom's DNA and dad's DNA. And then I'm going to use a 4th color that's going to represent a certain cell structure, which we'll talk about. So for me, I'm going to use black, red, blue, and then like a yellow orangey type color. Use whatever colors you want. If you've got those, great. So let's go ahead and I'm going to grab my black, so just my basic color. And in this cell, I'm going to start by just drawing a circle within the circle. And I'm going to draw the whole thing, and then we'll talk about what each component is. Next, I'm going to take my red, one of my parent DNA colors, and I'm just going to draw some squiggly lines in that circle there. Make sure they stay within that smaller circle. I'm going to take my blue, my other parent DNA color, and draw some more squiggles. And then I'm going to take my other color, the one that I said was going to represent a cell structure. For me, it's going to be this yellowy orange. And I'm just going to draw 2 stars over here. Okay, now that's everything that I need to draw in there, but let's label some things so we know what's going on. So first of all, I'm going to do this little squiggly bracket thing. You can do whatever you need to, but basically this little squiggly bracket is going to tell me that this whole big circle here, this is a cell, C-E-L-L, cell. Next, I want to label that smaller black circle that we drew. So I'm just going to draw an arrow right to that. And that, let me make sure I have some space to write here. I'm going to call that my nuclear membrane. Okay, nuclear membrane. And then I'm going to draw an arrow to the stuff that's inside of that nuclear membrane. And that is my chromatin, C-H-R-O-M-A-T-I-N, chromatin. And then finally, I'm going to label my little star things, which are centrioles, C-E-N-T-R-I-O-L-E-S, centrioles. Now, in real life, centrioles don't look like stars. In kind of a better reality, they kind of look like churros. If you're familiar with what churros look like, they're like little bundles of straws almost. That's what centrioles really look like, but that's kind of complicated to draw. So I just draw them like stars because it's much easier. So there you go. That's our first diagram of a phase in mitosis. Now, we've gotten our image drawn there. Let's go ahead and scroll over to here. You've got some blanks to fill in. So first off, we said DNA is in chromatin form. So those are the loosey-goosey squiggly lines that you drew in the nucleus. It's basically unraveled DNA strands. Okay. Also in interphase, DNA is copied to prepare for the cell to divide. So mitosis is when the actual division happens, but interphase is when the cell gets ready for that division. And finally, the cell spends most of its time in interphase. As we saw in that cell cycle, the majority of that cell cycle is spent in interphase, and mitosis really doesn't take up too much of the time of a cell's life. Okay, so let's go ahead and move on to our next stage, which is prophase. Again, I've already got it labeled here. You go ahead and label yours. And I'm going to zoom in a little bit more here. And I'm going to start out with my black pen. And I'm going to draw my nuclear membrane in there again, but this time I'm not going to draw it like a circle. This time I'm going to draw it like a dotted line. So I've got kind of a dotted circle in the middle of my cell. Next, I'm going to grab my one parent DNA color. Now this time I'm not going to draw squiggles in the nucleus. Instead, I'm going to draw 2 X's of each color. I'm going to draw a big and a little X of each color. So a big and a little red, and a big and a little blue. And it doesn't matter where you draw these, I just happen to have drawn them there. They can be anywhere within that dotted circle, okay? Next, I'm going to draw my centrioles. Last time when I drew them, I drew 2 stars next to each other on one side. This time, I'm going to draw one on each side of the cell, just like that. Okay, so there's our drawing for prophase. Let's talk a little bit more about that and take some notes. So first off, DNA condenses into chromosomes. So no longer is it chromatin, now we've got chromosomes. So those are those X-shaped things. Chromosomes are DNA in X-shaped form. I sometimes call it its traveling form. If it's all loosey-goosey and trying to split between cells, you may lose a chunk of it here or there. But when it condenses into chromosomes, it's more easy to get all of that DNA to where it needs to go. All right, so DNA condenses into chromosomes. The nuclear membrane dissolves. That's why we didn't draw it as a solid line. We drew it as a dotted line to show that it's kind of breaking apart and dissolving. And then the centrioles move to opposite sides of the cell. Like I said, we drew that. So we had in the first picture, we drew the 2 stars next to each other. This time we drew them on opposite sides of that cell, right? So that is prophase. Let's move on then to metaphase. Now this time we're not going to draw a nuclear membrane at all. Remember in the last step it was dissolving. Now in metaphase we don't have a nucleus at all. But if you remember, in the last video, I told you that meta, you should remember, means middle. And this is why. Remember, we have those 4 chromosomes, the one big red and the one little red, and then the one big and one little blue. What I'm going to do is draw those 4 chromosomes anywhere along. So I'll just do my big red there and my little red there. And then I'm going to draw my blues in there, a big blue and a little blue. Again, doesn't matter which order you draw them in, but they should be lined up single file down the middle of the cell. Remember, metaphase, middle, right? Meta means middle is what I'm going to have you. That's how I'm going to have you remember that. Next, let's draw those centrioles back in there. So again, they're just on opposite sides of the cell like this, okay? And then another important thing, and this is where the centrioles really kind of start playing their really important role. We're going to draw these little lines, and each little line, so each centriole is going to have 4 little lines coming from it, and each of those little lines is going to connect just to the middle of each chromosome. They'll kind of grab the, if you imagine the X shape, if you imagine it like 2 arms and 2 legs, it's going to grab it at the waist is what we say. So we're going to grab those lines are going to grab the chromosomes at the waist, okay? And that is our drawing of metaphase, right? So let's go ahead and look at this other part here. So like I said, in metaphase, our chromosomes line up in the middle of the cell, there we go. And then spindles form out of the centrioles and attach to chromosomes. So spindles are those lines that we drew. They're kind of like little grabby arms that the centrioles grow, and those spindles are what's really important as we divide our cell. So there are our notes and our picture for metaphase. Let's move on to anaphase. If you remember, I said that Ana, you should remember or relate to the word sister, okay? And here's why. Now, don't get ahead of me drawing stuff. When students get ahead, they end up doing this one wrong. So I'm going to start this time. Oops, that's not what I wanted. Let's go ahead and back that off, and we'll go to this. We're going to start by drawing our centrioles. And I'm actually going to draw my arms out just a little ways, my little spindle arms. I'm going to draw out just a little ways, not as far as they had gone before, because what those spindles have actually done is they've kind of contracted, and what they're doing is they actually pulled the chromosomes apart. That X-shaped chromosome is actually two copies of DNA attached together in the middle. And what it's doing is it's actually pulling those two halves apart. So what I'm going to do is actually draw, if you can imagine what a half of an X would look like. It kind of looks like a little arrow shape, okay? It's kind of like that. If you could imagine another, I'll draw it in there. This would be like a full X, right? But we don't want to draw that. We just want half an X over here, and I'm going to draw that other half of an X over there, okay? And I'm going to do that. I think I had the other little red down here, and I think I had, oops, that's the wrong one. I think that I had my big blue up here, my little blue down there. I may have put them in the wrong order. I don't know. But hopefully you can understand that that's what's going on. So remember, those little spindle arms actually pulled those chromosomes apart. All right? Kind of gnarly, right? The only note that you have for this one then is that the sister chromatids were split apart and they're moved to opposite sides of the cell. When they're attached at the middle, we call it a chromosome. When it's that X-shaped thing, we call that a chromosome. But once they're split apart, we call them sister chromatids because it's actually identical DNA. This side and this side are identical copies of DNA. And once we split them apart, they're two sister chromatids. They have the exact same information on them. So that's why I said anaphase. Remember, ana means sister. This is where we have those sister chromatids being split apart and they're being moved to opposite sides of the cell. Okay, there's our picture. Next, we've got telophase. You may notice that this is a very different shaped picture than what we've had before. So we're actually going to start with our black again here, and we're going to go back to that dotted nucleus thing like we had in prophase. And this time we're going to draw two of them because remember, we're making a copy, right? We're duplicating, we're splitting the cell in two. Okay. The reason it's pinched in like this, by the way, is if you can imagine having like a ball of silly putty and you want to tear it into two balls of silly putty, you probably kind of pinch in with your fingers, right? You pinch it in and then you split it apart. That's basically what the cell is doing here too. It's pinching in the middle and getting ready to split it apart. So we can see that pinching in with this funny shape here. So in each of those two sections, we've got our dotted nucleus. I'll go ahead and get my one parent DNA color. Now remember that we're still in that sister chromatid, right? So they're kind of still like a half of an X. So I've just got my two half X's of each color. So my four half X's all together in each of these, and it doesn't matter what arrangement you put them in, they're just kind of floating around anywhere, right? And then don't forget your centrioles. Each one should have one centriole in there somewhere. They still got to be hanging out there somewhere. So there is our picture for telophase. The only note I want you to write down for telophase is that in this stage, two new nuclei are formed. So nuclei is plural of nucleus. So we see that nucleus being formed again around those two separate copies of that DNA. Okay. So there's our picture. And then we're going to go down to cytokinesis, our last step, which some people don't even consider a part of mitosis, but it's important. So last stage, our nuclear membrane was coming back. This stage, it's all the way back. Okay. We're still going to draw the same stuff in the nucleus that we had before. So we've got our two half X's in our two different colors. And then each of them also has one centriole. So there is our completed picture for cytokinesis. And then the only note for cytokinesis, our very end, is that in this stage we have two new cells being formed. Two new cells are formed. Okay. So that is the whole process of mitosis. We started with a single cell that had a bunch of this chromatin in it. During interphase, it copied everything, and got ready for that division. Okay, and here's our whole process of division, and in the end we end up with 2 new cells. So we have that one cell becoming 2 identical copies. That's our process of mitosis. Hopefully this all makes sense. Hopefully you were able to get everything down on your notes. You're all set. If you have any questions about anything, don't hesitate to ask.